All right, it is now my pleasure to introduce Dr. Prince Kannankeril to give the next talk. Dr. Kannankeril is a professor of pediatrics at the Vanderbilt University Medical Center with faculty appointments in both the Vanderbilt Genetics Institute and the Vanderbilt Center for Arrhythmia Research and Therapeutics. He serves as the co-director for the Center for Pediatric Precision Medicine at Vanderbilt. His research program investigates genetic variants and their contribution to cardiovascular disease and clinical outcomes, and he will be speaking to us today about RYR2, big gene, too many phenotypes.
Well, thank you for that introduction. And I assume you'll let me know if you can't hear me or can't see my screen. The title of my talk was assigned, RYR2, big gene, too many phenotypes. I certainly agree with the first half of that. RYR2 is a big gene, but I don't think you can ever have too many phenotypes, right? Certainly RYR2 has yet to catch up with SCN5A, another one of my favorite genes. But our story today begins with this plant, which was discovered in the jungles of South America by the Danish botanist Martin Wall in the 1700s. This was a plant with insecticidal properties, and Martin named it after his friend John Ryan, who sent him many plant samples. And so we call this plant Ryannia speciosa. Now, the insecticidal molecule was identified in the 1940s, and because it came from the Ryannia plant, it was named ryanodine. And now you probably figured out where this is going, right? So the poison ryanodine actually targets the major calcium release channel from the endoplasmic or sarcoplasmic reticulum. And so we named that the ryanodine receptor, or RYR. In nanomolar concentrations, it locks the channel open, and that results in massive muscle contractions. And at micromolar concentrations, it closes the channel, resulting in paralysis. Either way, it's lethal for both insects and mammals, actually. Now, the RYRs are the largest known ion channels to date. They're more than 5,000 amino acids long, each encoded by genes that cover more than 100 exons. In humans, we have 3 isoforms, RYR1, 2, and 3. There's 65% identity in the genes between those 3 isoforms. RYR1 is the predominant form in skeletal muscle. RYR3 is the so-called brain isoform, because if you look in human tissues for RYR3, you see it predominantly in the brain, but actually when you look at brain, you find more RYR2 than RYR3. RYR2, of course, is expressed in cardiac muscle, but also in the cerebellum and the cerebral cortex, and at lower levels in multiple organs, including the stomach, kidney, adrenal glands, pancreatic beta cells, and others. So it really should be no surprise that there are multiple phenotypes associated with RYR2 mutations. The one we're most familiar with, of course, is CPVT, or catecholaminergic polymorphic ventricular tachycardia. This disease is characterized by a normal heart and a normal ECG at rest, as shown up here. But as these patients exercise, they develop a typical progression of ventricular ectopy, usually starting with single PVCs and then bigeminy and then couplets and then non-sustained, and in this case, sustained bidirectional ventricular tachycardia. And they're also at risk for ventricular fibrillation and sudden death. We've also come to recognize mild sinus bradycardia in many of these patients, as well as atrial arrhythmias. And more recently, we've recognized intellectual disability is fairly frequent, 8%, as documented by the International CPVT Registry led by Arthur Wild and colleagues. Now, slightly more than half of human patients with CPVT have RYR2 variants, and those that have been characterized typically have excessive calcium leak during stress. So some people call that a gain of function. That's not exactly correct, but it's a simple way to state excessive calcium leak during periods of stress. Now, there have been some non-CPVT related arrhythmias associated with RYR2. One of them is this phenomenon here called short-coupled polymorphic VT, or sometimes the short-coupled variant of Torsades-Dupont. This paper from Andrew Marks's group was a case report, really, of a young woman who had short-coupled PVCs and polymorphic ventricular tachycardia. So did her mother, and they both shared a mutation in RYR2 that actually caused excessive calcium leak, but under non-stress conditions. And then Minoru Hori's group in Japan actually looked at a series of patients with short-coupled torsade. They found several with RYR2 variants, but only one that was clearly pathogenic. And that one actually was associated with reduced calcium release, so a so-called loss of function. So sort of the opposite molecular mechanism that we see in CPVT was associated with short-coupled torsade in that particular patient. A somewhat similar story was recently reported by Mike Ackerman's group in the Amish community. These homozygous duplications in RYR2, and the phenotype sounded a lot like CPVT. These patients were dying suddenly during exercise or during swimming, but when you exercise them on a treadmill, they really had no ectopy or mild ectopy. And arrhythmias that were captured actually looked more like short coupled torsade, right? So just all of a sudden from sinus rhythm to polymorphic VT and then VF, not the typical progression that we see in CPVT. And a subsequent paper actually characterized the functional consequence of these duplications, and it was also a so-called loss of function or reduced calcium leak through RYR2. Now, one of the first papers that expanded the phenotype of RYR2 mutations was this one from Zahir Bouyan and Arthur Villa's group. And these were patients that actually had clinically recognizable CPVT, but they had much more severe sinus and AV node dysfunction. So it's probably hard to see this, but this patient at rest has complete atrial standstill and just a junctional rhythm. They also saw LV dysfunction and in some cases frank dilated cardiomyopathy associated with these variants. The other important piece of this was these were patients that had no mutations by standard sequencing techniques, and they only found a deletion in exon 3 by doing deletion duplication testing. And so this paper sort of highlighted the need to do that. And multiple groups have then documented this exon 3 deletion. It's a bit more common than we would necessarily think. So Minoru Hori's group in Japan, they re-examined their CPVT negative, or I should say their RYR2 negative patients with CPVT with deletion duplication analysis and found 8 mutation carriers that carried that exon 3 deletion. And interestingly, in 7 of them, they had LV non-compaction, as shown here. And this figure is actually taken from this paper from Thomas Roston and Shu Sanatani's group, who identified a patient with sort of a mild version of CPVT, but also LV non-compaction. That patient had a mutation that was associated with so-called loss of function, again, reduced calcium leak through RYR2. And this paper is more recent. I think the final version is still in press. Also associating cardiomyopathy phenotype here, so pretty clear LV dilation and cardiomyopathy with really just a mild arrhythmia phenotype in a family with a premature truncation in RYR2. Now, one of the points I wanted to make today very clear is that you may have a patient with a odd phenotype and an RYR2 mutation, and it doesn't automatically mean that those two are connected. And we know that thanks to some important work by Andrew Landstrom and Jeff Kim and the group at Baylor that sort of gave us the background noise, if you will, in RYR2. So when they looked at over 150 patients with CPVT, they found rare variants in RYR2 in 60%, and that's not unexpected. But when they looked at patients referred for whole exome sequencing, this is over 6,500 patients, 9% of them actually carried a rare variant in RYR2. And of course, the overwhelming majority of these patients would not have CPVT. And of course, when you look in public databases like EXAC, the Exome Aggregation Consortium, over 60,000 individuals, 6% have rare variants in RYR2. So just because a patient's referred to you with a variant doesn't automatically mean disease. Even variants that we would think would definitely be pathogenic. So Wayne Chen's group actually generated a mouse model with that exon 3 deletion that was previously associated with disease in humans. And interestingly, although of course no homozygote mice were born because that would be complete loss of function, the heterozygotes had no CPVT phenotype. Okay, so here's ECGs from mice after epinephrine and caffeine injection, which would normally recapitulate the ventricular arrhythmias, and you can see just sinus tachycardia in the mutant mice, same as you see in wild type mice. And so presumably this is because the wild type RYR2 is predominantly expressed in these mice with reduced expression of the mutant. And so I certainly have seen patients referred to me with RYR2 deletions expecting them to have CPVT, and they've had no phenotype whatsoever. So again, phenotyping is very important here. So some of the non-cardiac phenotypes associated with RYR2, this was really interesting work published in JCI looking at endocrine consequences of RYR2 mutations. So this is a figure where they took CPVT patients who had RYR2 mutations and compared to age and BMI matched controls showed that they have abnormal oral glucose tolerance tests with reduced insulin secretion. And there's a lot more work in this paper that I'm not showing on the slide, but they showed pretty definitively that leaky RYR2 results in impaired insulin secretion. There's a couple of papers on some diseases that I'm less interested in, and they're in journals that I don't particularly read. So if you're interested in either of these syndromes, you can read these papers as well. And I think I'm right at 10 minutes, so I'll just quickly introduce this last slide here. This was a really fascinating paper that I would encourage you to read on your own time. This is looking at mice, a mouse model of Alzheimer's disease, and these mice have increased neuronal hyperactivity. But Wayne Chen's group actually crossed them with a mouse model carrying an RYR2 mutation that reduced RYR2 open time, and they showed normalization of that hyperactivity. And in measurements of memory, sort of mice working through a maze, they actually showed that they could increase memory in this mouse model of Alzheimer's disease. So there may be some therapeutic targets involving RYR2 for Alzheimer's disease, probably because RYR2 is highly expressed in the brain. So I think I'm over my 10-minute time limit, so I will go ahead and stop there, and I'll be happy to take any questions.
All right, thank you very much. That was a really interesting talk. Again, if you have any questions, please post those in the app so that we can ask those to our speakers.
And I think we have maybe a few minutes for questions. One question, Prince, this might relate the last two talks together, but in a patient without much other phenotype, but maybe had a sudden cardiac arrest, when you get an RYR2 variant, how do you treat the significance of that?
Yeah, no, that's a great question. So, clearly patients who've had an arrest, you want to get a good exercise treadmill test on them. And sometimes you can't do that in the immediate post-arrest period. And, you've got to sort of let them leave the hospital with a diagnosis of idiopathic VF. And so I actually typically don't start a beta blocker right away if I don't have a clear indication. And I'd like to bring those patients back once they're fully recovered and they can do a really good treadmill. If they have any ectopy at all on exercise and an RYR2 mutation and they've had an arrest, I would probably treat them like a CPVT patient and then put them on a beta blocker. Those who, they have a VUS and RYR2 and they've had an arrest, but I can get a really good treadmill, exercise them fully, and they have no ectopy at all, then I still consider that a variant that hasn't associated with disease. I won't necessarily put them on a beta blocker. Obviously, if they have recurrent VF, then I might reconsider that. But I'd have to say most of my idiopathic VF patients, that have left with the diagnosis of idiopathic VF have not recurred, and so I haven't had that occasion.
Okay, great, thank you. Well, I think let me see. It looks like there's no other questions listed at this time, so I think we'll move on. Thank you very much, Prince. Appreciate it.
Great, thank you.